So I also want to thank Philippe for inviting me to participate in, together with Brett and Jean-Marc in this symposium. It's been a great pleasure to be hearing all these great talks. And I also want to thank Colette for doing all the hard work. So, we, in my lab, we're interested in cell-to-cell communication in bacteria and mainly in communities that are composed of multi-species. And we've been, as we've been hearing already, oops. Okay, so as we've been hearing already throughout this the day yesterday and even this morning, bacteria are, although they are unicellular organisms, they're like cells in a multicellular tissue. They can synchronize their gene expression and engage in multiple group behaviors. And we've heard already several, but bacteria can make things like production of bioluminescence in a synchronized matter, and they can produce biofilms, form biofilms, and even synchronize the formation, synchronize the production of virulence factors in many human pathogens. And this type of group behaviors are important for beneficial as well as hostile type of interactions. And as we've already heard in some of the talks, most of these group behaviors are regulated by quorum sensing, which relies in the production, secretion, and sensing of these small signal molecules, which are represented here by these blue triangles, which are called autoinducers, and which at low cell density, they diffuse away and are not sensed by the bacteria, but in high cell densities, especially in liquid cultures, they're sensed, and this sensing of these molecules usually triggers a response at the level of gene expression, enabling bacteria to regulate the group behaviors, such as the ones that I've just mentioned. And the type of group behaviors that are regulated by quorum sensing can be nicely illustrated by the most obvious example, which is bioluminescence from Vibrio fischeri, which is where quorum sensing was first discovered. And it's a really nice example because it's a typical example of something that is only productive when the cells are at high cell density, and this is what we typically see in the behaviors that are regulated by quorum sensing. And I have here another example from this Bactobacterium. It's a plant pathogen that produces, it secretes an array of plant cell wall degrading enzymes that are secreted to the extracellular media, or in this case to the extracellular potato that it's infecting and degrading the tissue. And again, this is a trait that is only productive when the cells are at high cell density. So this type of quorum sensing regulated behaviors, such as these ones that in the case of Bactobacterium can be a pathogen to several plants, or in the case of Vibrio fischeri, which is a symbiosis, there are cases of monospecific, they're really mainly driven by monospecific associations, and they're usually regulated by autoinducers, which are species-specific, where the species that are producing the molecules, they have a specific receptor to detect their own signal. And so we usually say that these type of signals are important for interspecies communication. But as I think most of people here know, most of the natural environments that we have are actually composed of many different species, and so it's not surprising that bacteria also have signals that allow them to sense the presence of other bacteria, and we think autoinducer 2 plays a major role in this process, as it was already mentioned by Johannes yesterday. Autoinducer 2 is produced by many different bacterial species that range from gram-positive as well as gram-negative bacteria, and these are just some of the examples of bacteria that produce autoinducer 2, and I've shown already a long time ago that you indeed can mix different species in the same test tube, and they can regulate gene expression via autoinducer2, so we can really say that it can foster at least interspecies interactions. We can discuss if it's really communication. So since the discovery of autoinducer2, it's been shown by many groups that autoinducer2 can regulate many of the typical current sensing behaviors that that some of them already mentioned. And so, for example, autoinducer2 regulates bioluminescence in Vibrio harvii, but it also regulates some of the behaviors that Melanie talked about yesterday in Vibrio cholera, like virulence and biofilm formation. As Johannes said yesterday, it can induce phage dispersal in bacteria like Enterococcus fecalis. It can regulate the formation of oral biofilms in Streptococcus gordonii and E. coli, and in Salmonella it seems to be regulating motility, and I'll tell you about how it also regulates interference of current sensing behaviors. So usually these studies on autoinducer2, they've been focusing in species-specific, in single-species settings in a test tube, and in our lab we are really interested in looking at interspecies, in the interspecies property of autoinducer2. So we are focusing in multi-species community, and I think I don't have to convince this audience that this is the most interesting interspecies community that we know of, the mammalian gastrointestinal tract. And this is a cartoon of how we, at the moment in our lab, visualize or imagine the mammalian gut to be, where hundreds of different bacterial species are interacting with each other through chemical molecules. It's also, we know that there are some species specific molecules. We know there's the autoinducer2, and it's also a good time to remind you that we think there's way more molecules than this one. I think we're going to hear about a new molecule today from some of the next speakers. And so we are interested in understanding specifically what autoinducer2 molecule is doing in this community, at least at the moment, because it's so far is the only molecule that we know that is being produced by very distantly related bacteria, such as gram-negatives and gram-positives, and so we think it's more likely to play a role in such a complex multi-species environment. So we already heard, and most of you know, the importance of the microbiota, especially the intestinal microbiota for the host health, and we've been focusing mainly in the colonization properties that bacteria-bacteria interactions that happen in the mammalian gut can have in order to protect the host, that lead to the protection of the host towards invading bacteria. So my student Ana Rita did a simple experiment to illustrate to you what we think of colonization resistance, but we'll hear more about it today in the afternoon section, actually from Barbel and Andreas and João, and they've been working on colonization resistance for much longer than me, but here's our simple colonization resistance experiment to remind you that what happens is when you have a very stable microbiota community in the gut, usually it's very hard to have a bacteria that can invade this community. And this is, in this case, is conventionally raised mice that have been oral gavaged with E. coli, and if we don't treat the microbiota, the E. coli levels drop very, very quickly, and E. coli cannot colonize the intestines. However, if we give streptomycin into drinking water into these mice. This causes a disruption of the gut microbiota, and also it's important to remind you that this E. coli is streptomycin resistance. So if we keep the antibiotic, the gut microbiota is going to be disturbed, and E. coli can colonize now the intestines at very high levels. These are colony-forming units of E. coli that were measured by plating the fecal samples of these mice. So colonization resistance is not about just having space for E. coli to invade in this case, even though when you give the antibiotic to these mice, we always see a drop in total bacterial load, and it's usually lower at day 4, and you see around like a tenfold drop in the bacterial load. But Anna-Rita did test that you can colonize E. coli to the same levels no matter if you give the E. coli at day 2, which is our usual protocol, or if you wait and you keep the streptomycin for 28 days, and now you introduce the E. coli here, where we've seen already the recovery of a microbiota. E. coli can still colonize the gut just fine. So what we usually see is a drop on the microbiota due to the antibiotics, and even if we keep the antibiotics, there is some recovery that usually is not different from untreated mice, but they are still, this microbiota composition is still very susceptible to the invasion of microbes, in this case to E. coli. And we know also by the work of others that this is due to the different composition that these mice have on their microbiota. And this is what we also characterize in the mice that we have in our facility. So untreated mice, when we analyze the microbiota composition by 16S in fecal samples, have the typical microbiota composition, which is mainly composed by 2 different phyla, the Firmicutes, which are here in blue, and the Bacteroidetes, which are here in red. A healthy mammalian gut has typically a 1-to-1 ratio of these 2 phyla, and it's been shown that these 2 phyla are quite important for many host functions, and that the proportion of the 2 is quite important. However, what happens during the streptomycin treatment that we do in mice by giving them streptomycin in drinking water is that very quickly we see an expansion of the Bacteroidetes phyla. So by the end of this almost 1 month treatment of antibiotic, the gut microbiota is really more than 90%, more than 95% composed by Bacteroidetes. And as you saw in the previous experiment, this type of composition makes the mice very susceptible to evading pathogens or evading microbes, even if they're not pathogens. So what Anna-Rita and Jessica Thompson, in collaboration with Carl Zobeda, who helped us in the microbiota analysis, decided to do was to ask if this change in microbiota composition that we observed during the streptomycin treatment could be influenced by the autoinduced HER2 current sensing signal, and if it would affect the dysbiosis outcome. So in order to ask this question, what we first needed to implement was a way to manipulate the autoinduced HER2 levels in the mouse gut, and for that we relied on a natural system that I had discovered already some time ago in E. coli that can manipulate the levels of autoinduced HER2. So what happens is that E. coli has a very unusual profile of autoinduced HER2 production and accumulation, and it produces autoinduced HER2 at the early stage of growth, but then it degrades this molecule at high cell density, and we have characterized the system quite well. It's an ABC transport system that takes this molecule and phosphorylates it and degrades it. And as a consequence, we see when there's high autoinducer 2, this operon is induced, and the autoinducer 2 is internalized and quickly degraded. And because we know this system quite well, we can construct different mutants that accumulate different levels of autoinducer 2. And these are the most important mutants for today's talk, which we have the wild type, the parent strain here in black that produces the signal and then degrades it. And then we have this blue mutant, which is a kinase that does not phosphorylate the signal, and it does not induce this LSR transporter, so the E. coli no longer degrades the autoinducer 2, so this strain accumulates high levels of the signal. And then we also have another strain here in red, another mutant here in red, which is a mutation in this repressor that is constitutively expressing this transporter, and this strain, I like to call it this AI2 scavenging strain. It's very efficient in removing the signal and depleting the signal from the environment. So we can manipulate the autoinducer 2 levels using these E. coli mutants, and we've done several experiments in test tubes with these mutants, but here's just one example. When we mix E. coli that does not degrade the signal with Vibrio harvii, Vibrio harvii turns on bioluminescence at high cell density by sensing autoinducer 2, and then if we pretend to turn off the lights, what we can see is that in the presence of the E. coli that doesn't degrade the signal, Vibrio harvii turns on bioluminescence, and the bioluminescence level will stay high. However, if it's co-cultured with the E. coli mutant that degrades the signal, then Vibrio harvii is tricked. It can no longer tell that it's at high cell density and turns off bioluminescence. So these were some of the experiments we did in the test tube to show that indeed we can use these E. coli mutants to manipulate autoinducer 2 levels and coron sensing behaviors in other species. And so what Jessica and Rita did was to use these similar mutants, and we engineered E. coli strains to accumulate different levels of autoinducer 2. These are the profiles they have in vitro, and they colonize germ-free mice, monocolonized with these different E. coli mutants, and we were already expecting that because this is a laboratory strain, that the only thing that regulates with autoinducer 2 is this mechanisms of degrading autoinducer 2. So as we expected, all the mice were colonized with the same levels of E. coli, no matter which mutant we were using, which is very helpful because now we can see if it was manipulating the autoinducer 2 levels in the intestines without being worried that they would colonize to different levels. And indeed, what if we have mice with no E. coli, these are our basal levels, and then the wild type is accumulating autoinducer 2. So it's actually, it seems that it's not degrading, it's just accumulating. The constitutively a non-degrading mutant, the LSRK mutant, which is here in blue, has high levels of signal, and then the repressor mutant, which is also not producing autoinducer 2, has very low levels of signal when we analyze cecum extracts of mice colonized with these E. coli strains. So in order to show that or to ask if this mutant could be used for scavenging autoinducer 2, we did some control experiments where we have, again, the mice with the LSRK strain that doesn't degrade the signal and it produces signal. And then we have mice colonized with the combination of the strain that produces the signal and the strain that degrades the signal. And you can see that the strain can indeed degrade signal produced by the other. And you can especially say this when we compare with this control, because here we have the LSRK strain that produces high levels of autoinducer 2 in combination with the strain that we call the control strain, because this strain does not degrade nor produces the signal. So there's high levels here, but low levels when we have the scavenging strain. So we can manipulate the autoinducer 2 levels using using these combinations of mutants. So then, as I said, we wanted to ask, can autoinducer 2 levels influence the composition of the gut microbiota during streptomycin treatment? So here, what they did was to give streptomycin in drinking water, and we can analyze the composition of the microbiota in the fecal samples, and we have 3 different groups of mice. One that is colonized with the LSRK mutant that does not degrade the signal, the one that removes the signal, and the control strain that neither produces nor removes the signal. And then we decided to analyze, to focus on the microbiota composition after 1 month of treatment, because we observed that at early stages of treatment there's a huge variation, but here the microbiota is already more stable. And when we analyze the 16S composition in these mice, we saw several OTUs that had differences when in the different mice colonized with the different strains. And here are some of the examples. So we have some cases where there's higher abundance in the group of mice that were colonized with the LSRK that accumulates high levels of autoinducer 2. And in some cases, there's lower abundance in this group. And what we saw is that there were several examples where the OTUs were different in the high AI2 group, but we never saw in this experiment a single OTU where there was difference between the control strain in comparison with the strain that was removing the signal. So in this experiment, it didn't seem that removing signal was making a difference, but increasing the AI2 signal was affecting some of the OTUs. And then if we looked in a principal component analysis at overall composition of the community in the different mice, these dots represent different mice, and in blue you have the community composition in the LSRK, the one that the mice that have high AI2 levels, and then in red we have the mice that were colonized with the strain that removes the signal, and then in black the control strain. And you can see that the mice that the composition of the microbiota in the mice that removes the signal or the control, they cluster together, and we saw that however, we saw that the mice that were colonized with the strain with the high AI2 levels did not cluster with this. They were different, and they actually don't cluster. They're more dispersed, and there's a stronger mouse-to-mouse variation in this group than in these two groups. As like yesterday, again, although I had read already Kevin's paper, but yesterday Kevin was reminding me how, you know, if we think there's cooperation, there's more dependence there's likely to be more dependence between different individuals. So we are doing time courses analysis and seeing if there's less stability in this microbiota where there's AI2 production, and if we think that AI2 is coordinating the interactions between this microbiota, maybe it's not so surprising that this group would have a less of a stable microbiota than in this case that maybe there's no cooperation. So when we analyze the microbiota composition at the phylum level, what was quite interesting to us was that we saw that the OTUs that belonged to the firmicutes, they were higher in the group that had high AI2 levels, and they were slightly lower and the bacteroidetes were actually slightly lower. So increasing autoinducer 2 led to changes in the microbiota at the composition level, and quite interesting to us, it favored the expansion of firmicutes And this is important because if you remember, I told you that the streptomycin is really impairing the firmicutes and it's favoring the expansion of the bacteroidetes. So what autoinducer 2 is doing here is seems to be favoring the colonization of the firmicutes. So we could interpret this as AI2 is ameliorating the effect of the antibiotics. So in a cartoon type of manner, what we have before treatment is a good balance of bacteroides and firmicutes, and then when we have streptomycin, the bacteroides dominate, and we know this is based on other people's work, we know this is quite harmful for the host, and here we were able to colonize mice that are producing AI2, and we saw a slight increase in the firmicutes. So it seems that AI2 is favoring the group that was more impaired during the antibiotic treatment. Just one more piece of information is that so this is another cartoon showing the main results, and so this for us is the first evidence that we can indeed manipulate the microbiota composition by manipulating autoinducer 2, and it also, this work also allowed us to start to identify OTUs and species that are likely to be responding to autoinducer 2, and we were quite excited by seeing that the ones that seemed to be favored by autoinducer 2 were the ones that belong to the firmicutes, and when we analyze the genomes of these bacteria, we see that most of the firmicutes produce have the Luxes gene, so therefore they're likely to produce autoinducer 2, whereas in the Bacteroidetes phylum, only 20%, less than 20% have the Luxes gene. So it seems that autoinducer 2 was favoring the producers, and this could induce a type of positive feedback loop, which is very common in current sensing systems. And overall, the work suggests that we could use AI2 to recover from this type of effect of the antibiotics. So in summary, we established a methodology that allows us to manipulate the interspecies communication in the mouse gut, and now we want to determine which are the species that can be hindered or promoted by autoinducer 2, and we want to identify what is direct and indirect, and okay, oops. So this work leads to the hypothesis that autoinducer 2 might help recover from the antibiotic dysbiosis, because what we think is that even though autoinducer 2 might be favoring the firmicutes, we have a much stronger pressure from the antibiotics that is likely to be killing them. So now we want to look at stopping at the antibiotic and looking at the recovery, and we have some models to start looking at the functions that are regulated by autoinducer 2, and we'll be looking for things like biofilm and other traits that are typically regulated by current sensing to see if that can be the explanation for the better colonization of the firmicutes in our model. So with that, I'll finish by acknowledging the people from the lab. I already mentioned that most of the work was done by Jessica Thompson and Rita, and here are collaborators and our funding agencies. And so I'll be ready for taking questions. Thank you.